Matt Might has been the director of the Hugh Call Precision Medicine Institute at the University of Alabama since 2017. At UAB, Matt is the Hugh Call Endowed Chair of Personalized Medicine, a professor of internal medicine, and a professor of computer science.
All right. Thank you. Well, it's a pleasure to be here and to present to you all. I know you're deeply interested in medical topics, but turns out I used to be one of you guys. I spent most of my life as a computer scientist, more specifically as a functional programmer and academic in functional programming languages. So I got to give you my standard disclaimer on this sort of stuff. It's slightly modified for this audience, and you might even consider it more of a confession in that sense. And this is a medical talk. I really mostly do, at least what I know how to do, is programming languages and functional programming. So actually, I even try to give a sense of what my work has been like over the years. And if you look at the split along object-oriented versus functional, it's almost all functional. In terms of the kinds of research I used to do. And if you look at the kind of stuff I published in up until quite recently, it was all PL and nothing in medicine. So all that is to say is that I really don't have any medical training, and if I give you medical advice in this talk, don't take it. Yeah, this is what happens if you try to remove ants from your backyard with gas and forget about what happens when you get an air-fuel mixture. I guess the ants are gone, so relocated or something like that. So I also want to make an observation. So this is a talk really about the fusion of CS and biology. This fusion is not even remotely new. It's been going on really since the very beginning. Some of you might be familiar with the famous biologist Alan Turing. So actually, this is not even, this is true. He's actually a biologist. You just didn't realize it. In fact, to prove it, go to Google Scholar and look at his publications. His top publication is in biology. More citations than everything else. It's his one paper in biology, but it's a foundational paper in biology. And he's using computational modeling, essentially reaction diffusion systems, to show how do complex organisms evolve from single cells. How do you get the interesting patterns you see in nature, like, say, the stripes on zebra or the shapes of a leaf? Well, even before we knew what DNA was, he got it right. He found the model that actually predicts how this works in nature. And so he's still widely regarded within mathematical biology as one of the founders of the field. So I'm going to make some claims today. I will claim that data is the greatest drug of the 21st century. I really do think that when we look back from the end of the century, we will believe that this was the best thing we ever used for improvement in human health. I'll claim that this emerging field of precision medicine in which I now work delivers data as a drug. That's really what we do. And that's because precision medicine is a process. It's a step-by-step process. So I'll present an algorithm for doing precision medicine. And I'm also going to make another claim. For those of you who've spent enough time drinking the FP Kool-Aid to play with proofs and inference rules and things like that, I'm also going to claim that for precision medicine to work in practice, you have to generate proofs. And by that I mean every time you make a recommendation to a physician, you have to tell them why you think it's a good idea. You can't just say, my algorithm said so. Turns out they will not take your advice. So there's a role for a lot of FP technology in precision medicine. So what is precision medicine? Well, I think it really took off back in 2015 when Barack Obama launched the Precision Medicine Initiative. And this was designed to lay the scientific foundation for the field as a whole. And he defined it very simply back then as precision medicine is all about delivering the right drug to the right patient at the right time. So, and that's a good working definition even today. Now, after that, there's this explosion of terms that tried to claim the mantle of precision medicine. Like everybody who said, I do precision medicine, this is what it is to me. I spent a lot of my time since then trying to unify this under a common definition. And I do think that this definition, the use of data, particularly big data, to optimize health counts, is what precision medicine is really all about. So for me, precision medicine is an optimization process. And some physicians disagree with this. They're like, well, but I always try to give my patient the best medication. And that's what I've been doing this entire time. And what I tell them is, yeah, but you didn't have as much data now then as you do now. And you didn't have so much data that in order to find the optimum, you have to use a computer to do it. So precision medicine is a natural consequence of huge amounts of data becoming available on a per patient basis and the subsequent computational demands for finding the right thing to do with that data. So it is a process, goes from a patient ultimately into recommendations for them. It's either a pill or a procedure of some kind, but it starts with their data. And the piece of data that really kicked off precision medicine, it really is the human genome. This became a clinically accessible entity, a few years ago, and people started to grapple with the consequences of what it means to have the blueprint for a human being when you treat them. For a medical condition. But all forms of data ultimately count as inputs to this optimization process. And even the data that was already laying around at the time, like the electronic health record, even that was being poorly utilized, and honestly still is being poorly utilized in day-to-day healthcare right now. But everything counts. Fitbit data, Apple Watch data, social media data. Turns out you can make inferences, reasonably accurate inferences about mental health from a person's, in some cases, from people's social media data. So I think all of that counts for constructing this constrained optimization problem, where you get to modulate things like treatment and lifestyle and maybe even environment in some cases to try to find whatever brings a person to their optimal state of health. So a lot of precision medicine is trying to modulate these two factors, treatment and lifestyle, to find what that person actually needs. And I'll tell you right now that a lot of the time in practice, we don't jump all the way to an answer. In many cases, what we recommend is not a pill, but some kind of scientific procedure where we say, you have to go run an experiment because we don't have enough data, but we are pretty sure if you did this, you would have enough data, or you'd be closer to enough data to answer this question in a meaningful way. And I've also been claiming more recently that you also have to give a proof along with whatever recommendation you make. And this is from direct practice with doing this with real patients, where if we try to tell their physician that you should give them this pill and then don't tell them why, they go, no, I'm not going to do that because I'm legally liable for that. I have to know why I should do this for a patient. And I think one of the pitfalls of a lot of machine learning systems like Watson, when they try to make recommendations for patients, is that they don't have a proof that comes with it. It really is, just this is what the neural net says, and physicians don't like that. So in practice, I'm going to tell you for how this works in practice anyway. I'll give you my personal journey through precision medicine. Like, why is it that a computer scientist is giving you this talk anyway? And I'll use that to illustrate this algorithm, large paths of it, and then I'll generalize it and show you what it looks like for other patients too. So this really begins for me with my son Bertrand. So this is him back when he was a year and a half. He's 12 years old today, but life was rough for him when he started out because things were wrong and we didn't know what was going on. We were trapped in a place that a lot of patients call Undiagnosed Island, where you've got a child or a family member, they're suffering from some condition and you just can't figure it out. In Bertrand's case, we suspected it was a genetic disorder, and what it manifested as for him was seizures, in his case hundreds per day, extreme developmental delay, a movement disorder, and for some reason an inability to cry tears. So he could cry, like even through the emotions of crying, but he would never make liquid tears. And on a day-to-day basis, honestly, that was the worst thing to deal with. If you have chronically dry eyes, if you make no tears, it's like your eyes act like sandpaper on your cornea, and they were scraping off his cornea, and he was going functionally blind. He had eye surgeries to correct the infections in his eyes, and it actually had reached the point shortly before his diagnosis that said, look, we want to sew his eyes shut just to protect what remaining vision he has. So on a day-to-day basis, that's what was really, causing problems for us. So that was our life for 4 years, for 48 months, trapped on that undiagnosed island. And we knew this whole time, and we'd speculated based on everything we saw that there had to be some kind of bug in Bertrand's code. There had to be a genetic mutation driving this disease. And the question was, well, where is it? And how do you find that? So what I'll do now is give you a computer scientist's introduction to the relevant parts of biology. So for Bertrand, we had to get a source code. We had to look into his genome and try to figure out what exactly was happening here. And so you can think of the human genome as a very long string. It really is. It is exactly a string. It's 6.5 billion characters long, and with a genetic disease, you're looking for one or two mutations in that string. So the challenge is to find that typo in that big-ass string. And so DNA is a string, and it's got only 4 letters in it. This is the alphabet, A, T, C, and G. And these are encoded as nucleotides within DNA. And DNA, or subunits within it called genes, get transcribed into RNA, and transliterated or translated into RNA, and then transcribed into proteins, translated into proteins. And they do this 3 nucleotides at a time. So for every 3 nucleotides that you get a different amino acid. So you get this chain of amino acids formed from a chain of nucleotides. These fold under some biochemical properties into the resulting protein. And then there might be some modifications that happen after the translation of the protein. These are called post-translational modifications like glycosylation or phosphorylation. But that more or less is how you get a protein. And that's a lot of biology right there. That's like the central dogma of biology. And the key thing to remember is that the genome has a syntax, and more than that, it's actually DNA is really just an instruction set. So for each 3-letter sequence within a gene, that's an instruction. So if you have this little microgene right here, you can break it down, you can parse it, and you can make sense of what it's telling you to do. So ATG, so you always begin your program with an ATG for the insert a methionine. So this is the start of building a protein. The next instruction, GCC, you may know what this does. Well, it compiles C. But in our world and in biology, GCC really means insert an alanine. So that's, and then the last thing is actually TGA. There's a few ways to do this. It means stop construction of this protein. So this is what this little program actually means. It's begin construction, insert a methionine, insert an alanine, done. And then you'll get this little 2 amino acid peptide, and it won't really fold much because there's only 2 of them, but they'll then orient themselves in space. And if you have more amino acids, they just start folding according to the properties of their side chains. And that's how you get proteins. So mutations are modifications in these strings. So you can change GCC to GAC, and this changes the program. So instead of inserting an alanine, now you're inserting an aspartic acid. A lot of mutations are actually benign. We all have tons of mutations that make us different from the standard reference genome. Some will destroy function. Some will actually increase function, which may be good or might be bad. And some will change function entirely. So they can make it toxic. They might actually wipe it out. It just depends on the particular mutation. But this is the team that ultimately helped us look into Bertrand's genome. There was a new technology that came out that made this suddenly practical almost overnight. And it was done on a research basis. It's called exome sequencing. With exome sequencing, you look at the 2% of the human genome that actually encodes proteins. So it turns out only 2% encodes all the proteins you have. And almost all the changes that drive human disease occur in that region, at least major human disease. If you want to change one bit and get the most change to a human being, that's the region you change it in. So we tend to find genetic disorders clustered in this 2% of the genome called the exome. And suddenly it became possible to sequence only the exome. So you got this massive cost reduction overnight in genomic sequencing. So when we looked inside Bertrand's exomes, we found that he had 2 mutations in a gene called NGLY1 that destroyed the function of this gene entirely. So, and these are basically just syntax errors in the genome. One was a stop instruction got inserted in the middle of this gene, so you get half the protein out and it just says eject. And so you get a broken protein. The other one actually deleted a character, and so it shifted all those instructions off by 1 character. So now everything after the shift was just garbage. I mean, it still had a meaning in terms of amino acids, but they were nothing like the original amino acids that were there. So they really are just syntax errors, and you can spot them like syntax errors. So these are the easiest damaging mutations to find in a human genome. So it's like this thing just stood out like bright red. It's like there's probably something bad that just happened here because almost certainly this protein is gone, and it was. And then they said, oh, by the way, we think we found the cause, and if this is it, he's the first patient to ever have this disease. No one's ever seen this disease before. So he's the only one we know of in the world. He really is an N of 1. And that was about 7 years ago now, sitting in a lab at Duke getting that answer. Of course, I said, well, how do you know that these mutations drive the disease? And then this became a process which we now repeat a lot these days for many patients, which is called variant interpretation, where you say, okay, this mutation has happened. How do you know that this really drives what we're seeing in the patient? So we took essentially all the aggregated medical records we had for Bertrand, all these data points, and we tried to map them back onto the function of this missing gene to see if this explained what we were seeing inside Bertrand. So what this particular gene, NGLY1, does, and this won't mean anything to you, and I guarantee you it didn't mean anything to me all those years ago, was it's an enzyme that is responsible for deglycosylating misfolded end-linked glycoproteins after they have been retrotranslocated from the endoplasmic reticulum into the cytosol. So yeah, I mean, that's the sentence I heard 7 years ago, and it's like my functional programming brain went, what? What do any of those words mean? So I understand it now, and it's become part of my life's work to understand it. But in a nutshell, Bertrand is missing a recycling enzyme. There's a certain kind of cellular waste called misfolded glycoproteins, and this thing was suddenly gone. And so if that thing goes away, the trash piles up. It's really as simple as that. So that's really what it was. They said, we're pretty sure this is the cause of the disease. Everything seems to line up. And then, of course, they said, but there's nothing you can do about this. I said, he's the first patient. There's no drug companies working on this. They said, we can't even give you a prognosis. I mean, no one knows what this disease will do to him at this point. But I just disagreed. Maybe it's, my nature as a scientist to refuse to believe in non-actionability. But I said everything fundamentally is actionable. It may not be clinically actionable right away, but there's always something you can do. And that something is always going to be science. You can always run experiments and find out more. And maybe that will lead you to some actionable insights for the patient. So in precision medicine, for a lot of the cases we deal with, we don't have a drug right away, but we can always prescribe an experiment. And we do focus on that pretty aggressively. So that's 4 years. 4 years down into the Odyssey, we finally get an answer that he has NGLY1 deficiency. And I'll start to abbreviate some of the stuff that happened next. I started turning a lot of my intellect. So I'd learned a lot of genetics at that point. At that point, I dove into glycobiology to try to understand what was really happening inside Bertrand. I also knew that there was no way we were going to have one family take on an entire disease. I knew we had to find other patients that had it. So I ended up writing a blog post. This is the blog post that I wrote. It was really designed to do two things. It had to go viral, and it had to rank very highly in Google search results. So I did some SEO on this. And what I wanted to do with this was I wanted to bring other patients to us. I wanted them to find this when they were typing in things like lack of tears. I wanted them to land on this page and figure out that they had the same disease. So the title, picture of Liam Neeson, that's all the clickbaity stuff you need to make it go viral, because the internet is just so sadly predictable in certain ways. And it actually worked. It really did go viral. And I sometimes wonder if I put a picture, like a cat picture right there, would that have done just as well? And it probably would have. I mean, honestly, it's just how these things work. So, that post spawned a number of things. It led to an article in the New Yorker. And it led to me finding patients all over the world. So we found about 70 patients for this disease all across the world in the last 7 years. And you can make principled estimates of how many there are total, based on the frequency of the pathogenic alleles in these population databases. And I estimate there's about 500. There should be 500 patients that have this disease somewhere around the planet. And things started snowballing from here. So the New Yorker article led to my involvement in something called the Undiagnosed Diseases Network, where the goal is just a brand new NIH project, national project, to scale up what we'd just done for Bertrand, but for literally the entire country. And this is a project that's still going. I'm actually now one of the co-PIs on the project. Finding all the patients led to interest from NIH. They said, we want to do a study on all these patients. And so all the patients with this disease go to NIH once a year for 1 week at a time to do really in-depth study on their biology. Now, if you start to combine everything that we learned from the patients and from the biology, we're able to start making predictions. At the same time, my engagement with the Undiagnosed Diseases Network led to a faculty position at the Harvard Medical School. I still haven't told them I have no formal medical training beyond a C in 6th grade biology, but for some reason that doesn't bother them. So the therapeutic predictions where things started to get really interesting. So this was 2 years post-diagnosis here. I was able to make a principled calculation that something might be beneficial for Bertrand. And the way I did it was I said, okay, in the absence of this enzyme, some things are going to pile up. We know what those are. Those are these misfolded glycoproteins. But is there anything Bertrand's missing as a consequence of not being able to recycle these things? And there was some answers there too. And so there is even a computational way to do this, but you can do it with pencil and paper too, which is what I did at the time. I said, it looks like the sugars attached to these glycoproteins aren't going to get properly recycled. And what if this recycling process isn't just to get rid of the trash? What if it's actually an essential source of these sugars too in this particular part of the cell? And it turns out it is. So it turns out you're deficient in something called N-acetylglucosamine if you don't have NGLY1. Your cells are about 90% lower in this than they otherwise would be. And this is actually a pretty important metabolite. It has a lot of functions inside the cell. And here, Bertrand was missing it. And so I did what my computer scientist self knew how to do at the time, which is to use Google. The name meant nothing to me. You have to understand. It was like a symbol. It's just a random symbol, variable name. It's like foo. And I type it in and realize you can buy it on Amazon. So the logic is, okay, Bertrand's missing this. You can buy it on Amazon. So I bought it on Amazon. It shows up 2 days later, thanks to Prime. And then there's like, oh God, it's like a Star Trek level ethical problem now. Do I give it to him? So the first thing I had to do was, okay, let's just see if it's somewhat safe. And so I ate an entire bag of it in one sitting. And I woke up the next morning alive. And I said, well, FDA phase 1 safety testing is complete. Yeah, but then they were still like, wow, do I give it to Bertrand? I don't know. So I did give it to him after, because occasionally when he would have these severe hospitalizations, I remember a moment sitting by his bedside of the hospital thinking, my God, if this thing was sitting on my shelf and I never found out what it could have done for him, and this is the end for him in the hospital, I don't know how to forgive myself as a parent. And so once he was out and stable and healthy again, we did a little N of 1 trial for Bertrand. And I didn't know what to expect, because there's a lot of components to this disease. It's hard to predict what downstream mechanism this might actually impact. But what it did for Bertrand was after about 3 days on this compound, I was making him breakfast, went up to feed him breakfast, and I looked over into his bed, and he was crying. And it wasn't just like crying. He was actually making tears, for the very first time in his life. And I was just shocked. I just never had seen this before. And remember, his eyes were just in terrible shape. From this chronic lack of tears, and so suddenly there's tears rolling down his cheeks, and I go, "Oh my God, this is different." The only thing that changed recently was that supplement, and so I thought, well, maybe that's what did it. Turns out it is. But at that point, I did what I think any normal parent would do when they see their child cry for the first time. I collected his tears and I packed them on dry ice, and I shipped them to a lab in California for analysis. I mean, I really did that. In fact, I collected his first 80 tears, I think, and sent them all away. So those first tears ended up swelling into an ocean of science for the disorder as a whole. I mean, we learned so much about the biology of this disease from making him cry. It was really astonishing. You all get that that's a good thing, but when you give this talk or something like this to biologists, they go, oh, that's great. It's nice you can help people, but we're biologists, and we have different concerns. We want to know if it works in flies. So, okay, let's find out. So we genetically engineered flies to have the same disease. Turns out it's pretty bad for flies. About 80% of them die without this gene. And then we go ahead, we fed them this sugar, because that's what it is. It's a weird sugar is what it is ultimately. And when they're raised on the sugar and maintained on this sugar, their survival rate shoots up to about 80-90%. So it makes a big difference for flies. I don't know if they cry or not. I don't know if flies have tears, but they're alive. That's a big difference for them. I was inspired at that point to see, okay, I've been able to help Bertrand somewhat. Can I start to help others, and how much further can I go? So around that time, I ended up co-founding a company that does drug screens specifically for ion channel-driven epilepsies, where we try to find an existing drug to work for them. That ended up being so successful that after 2.5 years, we were acquired by Q State Biosciences. That was last year. And then I actually ended up getting a grant somehow to work on novel therapeutics development for NGLI1 deficiency. This was collaborators at the time back at the University of Utah. And I'll short circuit what we did here. We played around with planarian worms. A lot died in the conduct of this research. I'm sorry to say that. But we learned a lot of things about NGLI1 biology yet again. In particular, we found that if you take a second gene out of worms that are already missing NGLI1, they survive, and you can sort of rescue the major features of the disease for these worms. And that gave us a drug target. By realizing that it was better to have 2 genes missing than 1 missing, we could say, okay, well, how do we chemically disable that second gene in Bertrand? And that meant we had to find an inhibitor. So even if you don't know anything about drugs, you'll see on the labels of bottles that things are inhibitors for this or that or the other. A lot of drugs are inhibitors. They inhibit some mechanism you have inside of you. Well, we found a mechanism that we wanted to inhibit inside of Bertrand, and it's a gene called NGase. So turned into computational screening. We did what are called docking simulations. And the shortest explanation of this is it's exactly what video game programmers do with physics. So it turns out molecular dynamics, if you want to precisely simulate a protein, 3-dimensionally physically interacting with some other molecule, that's incredibly expensive. In the same way that simulating physics in a video game is incredibly expensive. So video game programmers make all sorts of shortcuts and approximations and cheats and hacks, and that's what drug designers do as well. So they say, okay, let's assume the protein is a rigid structure, first of all. So now things just start to get a lot easier. And you just say, is there any small molecule out there that is inverse in shape and charge to the catalytic domain on this protein? So that's a much more tractable problem to solve. In fact, we were able to screen 200,000 different structures in this fashion over the course of a few days. We found 70 compounds that looked like they were inverse in shape and charge to that catalytic domain. 14 of them happened to already be FDA approved. And this gets to a major feature of precision medicine, which is trying to leverage existing approved drugs and really the side effects of those drugs to do something you want. So when we took them to the lab, we tested them using mass spectrometry, and one of them actually worked, and that was Prevacid. So Prevacid is for acid reflux. Some people in this room take it. It's a widely used drug. Its designation is a proton pump inhibitor. So it is a proton pump inhibitor. That's what it does. However, this entire time, it had this hidden side effect that no one knew about. It's also an NGase inhibitor. And what Bertrand needs is to inhibit NGase. And so Prevacid looks like a promising therapy for him. So he's been on that for a few years. We've seen significant gains in that time period for him in terms of his development and communication capacity. And then things got sort of exponentially weirder for me over time. So I was giving a lecture at the med school at Stanford on a Monday or Tuesday, I think it was, and I got a random email saying, "Hey, can you come to the White House by Friday?" I thought, yeah, I guess sure, why not? I mean, I guess I'll do that. So I showed up at the White House on Friday. And as I'm landing the night before, they sent me these really cryptic instructions like, yeah, just like let yourself in and, wander around, do whatever you want, I guess. And then just go to the portrait of JFK at 10:30 and somebody will meet you there. All right, fine. So I did that. Showed up at the White House, went through security, said, put your phones away. So I hid it in my coat pocket, kept taking pictures. That's the basement. That's the Great Hall. And then I just gave myself a tour. It's a basement. It's still like the library down there. It's a sort of surrender from, literally, I just had an hour to just do whatever I wanted. It's weird. It's the China. I found it. I found the portrait of JFK. So I just waited there at 10:30, and sure enough, somebody opened another door and said, come right this way. And I was in the suite of what I now know are the staterooms. I just wandered around there for a while. Again, give myself a tour, taking pictures. And then at some point, one of the guards is like, hey, you can use the furniture. You don't have to stand this entire time. I'm like, well, if I can use the furniture, I will use the furniture. I'm going to sit on every piece of furniture in this place. And so I did. Sit on all the things. You just don't let computer scientists in the White House. We're very weird people. We will test the limits of all the rules you give us. So suddenly they're like, okay, give us your cell phone. Honestly, it wasn't a problem because then this guy walked in and he said, okay, so I read your story in the New Yorker and I was curious if you'd be willing to help out on this initiative I want to set up called the Precision Medicine Initiative. So I think what I'd really like to do is scale up what you've done for Bertrand, but for everybody. And I said, okay, I'll do it. What else do you say, honestly? And that for me was the start of a 3-year engagement with the White House. So one of the very first things I did was put together a white paper describing the architecture of this forthcoming scientific initiative to build a scientific foundation for all of precision medicine. Today, that program is known as the All of Us initiative. And what it's doing is it's collecting 1 million genomes and medical records for Americans so that we can build essentially a correlation between mutations and impact on health. That's really what we're trying to do because it turns out it's really hard to figure out what mutations mean. If you go down to the molecular and try to extrapolate what that means on the other side for a human being. So you can think of this as building the Rosetta Stone of the human genome. Where on one side we have genomes and mutations, on the other side we have medical records. So again, we can draw these correlations. I spent a lot of time with the Million Veterans Program, which had already collected 500,000 biospecimens at that point and had 500,000 genotypes, if not full genomes. So there was a lot of data there to play with. I ended up launching a small program within the Precision Medicine Initiative called PEPMA, which is the Patient Empowered Precision Medicine Alliance. And the whole goal of this was just to repeat what we'd done for Bertrand, but several times over for other disorders. Just want to quickly identify treatments, and we did it for about 5. The whole point of this was we wanted to have some things we could point to, say, look, it's not just Bertrand that's done this. There's we've been able to do it again. And so we did it for 5 different ion channel-driven epilepsies, plus 1 ubiquitination disorder. We did find treatments in less than 12 months. So when I went back for one of my final briefings with Obama, I was able to say, yeah, look at these. We did it again. And so I think that your vision for precision medicine is quite real. I think we're going to pull this off. You may recall there was an election. And I actually stayed at the White House. So I got an invitation from the transition team led by Pence to stay on because they didn't have enough scientists. So I did. It was interesting. So I did that for about a year. Wow, and it was a very interesting year. I'll put it that way. And after a year, I quit. It just got weird enough. Yeah, I think legally, if you look at the documents I signed on the way out, I think all I can tell you about that time period with normal blood alcohol levels is that it was interesting on a day-to-day basis. So, and I infer from news reports that it more or less is much the same these days. So then I got an unbelievable offer from UAB. They said, we want to build a whole institute around precision medicine. Would you be willing to come down here and build it from scratch and achieve, with the White House has been trying to set this up at a national level, lay this foundation, and here suddenly was the opportunity to go use that foundation and take it all the way into the clinic in a scalable way. So that was the goal, to scale up everything but on the ground in the health system at UAB for those patients. So how do you do this in practice? How do you do precision medicine over and over again? Well, a lot of it is drug repurposing. Developing drugs developing a drug from scratch for a single patient is just not even close to tractable. It costs like $2.6 billion on average to make a drug. But it's very easy to see if something else out there actually works. And that's a lot of what you do with this. Is you take the patient, you extract from them a model of their disease. It could be even their own cells. And you find a way to do what's called phenotyping. You do scientific experiments or assays to observe the disease in that model. Now, once you have a signature, this phenotype for this disease, then you can start testing drugs on it and see if anything out there rescues it so that you have the model going back to what looks like a healthy state. And then the hope is that whatever worked on the model also works in a human being. And sometimes it does. Sometimes it's not potent enough, and so you have to do medicinal chemistry. But there are lots of ways to deal with going from what works in a model up to what works in a human being. We've done this for other things like SCN8A epilepsy, where we really did screen 2,000 drugs against a cellular model of this epilepsy. If you want to know more about it, I'm not going to go into the biology stuff here. You can read about it. We've published it since then, and the kinds of stuff we did. One thing I will show you, though, is the cutting edge of this kind of screening. So this is the company that bought my company, and they do just like the most ridiculous stuff ever. So they modify neurons with 2 different genes. They put 2 genes into the neurons. One of them is sensitive to blue light and excites the neuron. The other one emits red light when it's excited. So if you raise a population of neurons from some patient, which you can derive from their skin, and put these genes in, you can grow these neural networks up, and then you can poke their brain, their little externalized brain for these patients, and watch what happens. So you are watching now what happens when you excite manually, by choice, an individual neuron. You can watch the propagation of signal to neighboring neurons with just extraordinary resolution. So this is something we can do now on a patient-by-patient basis. It's not cheap yet, but we can do it. And we can do this to build really high-resolution models of almost any neurological disorder that has a genetic component to it so far. So this works on ALS and Alzheimer's and a lot of epilepsies very well. And then we can start dropping drugs on it and see if anything rescues the phenotype that we observe. Another way to do it, and this is the way that gets back into functional programming, is to use logic programming. And some of you probably know Will Bird. He's the developer of miniKanren, and he's down faculty at UAB with me now. And we've been using logic programming to do drug repurposing. And so really the way this happens is we're just applying good old-fashioned logic to all the biomedical knowledge that we can get our hands on. So the shorthand version, the way I describe it is, take old-school logic, combined with high-speed automated reasoning to achieve superhuman-like deduction with clinical insight. That's what we're trying to achieve with miniKanren. And so you can ask it very low-level questions. You can say, what's an inhibitor for this particular mechanism? Or you can ask it very high-level questions, like what might be a treatment for this disease? And so, for example, if you ask it about, say, overactive bladder, it can come up with 145 different suggestions for what to try. If you ask it for a disease that has no known treatments at all, like Fanconi's anemia, it actually generated 10 of them, the top of which was xylitol, the kind of stuff that you actually find in chewing gum. And the reason it said xylitol looked like a compelling treatment for this disease is that there's a logical explanation. It says, xylitol inhibits alcohol dehydrogenase. Alcohol dehydrogenase produces reactive aldehydes. Reactive aldehydes cause double-stranded breaks in DNA. Fanconi's anemia patients cannot repair double-stranded breaks. So there's this chain all the way through that says this is why xylitol might work. And I'm not even kidding. I'm talking to people like Wrigley's research about trials of chewing gum to prevent head and neck cancer in Fanconi's anemia. So how do we do this? How do we build this tool? Well, we did a lot of reading. Obviously not us. It's all natural language processing. So one of the first data sets to come in was all the abstracts published in medicine, which is about 30 million of them. And we synthesized this into a knowledge graph so that the nodes are things like genes and drugs and symptoms and diseases, and then you have relationships between them. So, for example, there's an edge in there now that says Prevacid inhibits NGAs. That's something we learned as a consequence of my research. And now you just have a big old knowledge graph, and you can query it using logically meaningful relationships so that you can say, what is the biological significance of this concept relative to this concept? And you can prune paths that are sort of just paths that don't have any real biological meaning. So the core, the core relations inside of miniKanren are things like, this drug inhibits this enzyme, this disease manifests with this symptom, this gene or this drug increases this other gene, and I think there's about 16 of them that we initially started off with. I think we have 80 different relationships between concepts now. And from this, you can form artificial relations. So may treat is something where it says this drug may treat this disease if this gene causes this disease, and this disease is known to increase this gene, the expression of this gene, and this drug decreases that gene. So this is a very, very crude example of a way to look at a synthetic predicate. And of course, you can represent this as something like an inference rule. So we do think about a lot of what we do as an inference rule, the kind of which you might play around with sometimes in PL. And then, in general, we have strategies for how to find novel relationships. So, for example, if blue increases red and red decreases green, you can infer that by increasing a decreaser, blue will decrease green. So even if there's no known target for going after green, if we can pass through another gene on the way, sometimes we can find an indirect way to hit that target. And in fact, this turns out to be tremendously useful for finding new uses of existing drugs. And again, we can represent that as inference rules as well. So why do proofs really matter? I mean, they're helpful for actually finding these things, but it turns out proofs themselves matter because physicians don't believe you unless you give them one. Now, they're not looking for a proof tree. They want something rendered in English text that they can comprehend, and what they really want is a proof. So we've embodied this at UAB in the form of a research consultation service. And our goal at the service is just to always find the next step for any given patient that reaches out on their diagnostic or therapeutic odyssey. So we do follow a step-by-step process, which you can think of as an algorithm. So just to show you the algorithm in high speed in flowchart form, I have it right here. And then I'll zoom in on the core of this algorithm in just a second. But it's really broken into two parts. There's a diagnostic part where you're trying to zoom in on the core mechanism, and there's a therapeutic part where you're saying, okay, as a consequence of this mutation, what has happened to some molecular mechanism in this patient, and how do we compensate for that? The goal is to ultimately end up doing an N-of-1 clinical trial where you measure the impact to the patient on that drug. So this is the heart of the algorithm right here, and it really explains almost everything that we do. We find the core mechanism, and then we ask what's happened to it. And this applies beyond genetic disease. Almost every disease has mechanisms involved in some way. We say, is it overactive? In which case, inhibit it. Is it underactive? In which case, activate it. Is it absent? In which case, compensate for it. Or is it toxic? In which case, eliminate it. And we've got big game plans behind each of these depending on which direction we go with any given patient. And we have seen lots of patients. So we started this program about a year and a half ago where patients can just reach out and say, hey, I got an ultra rare disease, or I don't know what to do at this point. Can you help me? And people do reach out. So the question I've been asking over and over again is, how do we scale this up? And clearly automation is a big part of this. There's no way we can do this with manual intervention the entire way. But it turns out it's not all automation. Actually, we do have a secret ingredient that really makes this process go, and that actually turns out to be people. In particular, undergraduates. So we have a good team of undergraduates, and undergrads specifically for a couple reasons. One is that they're really cheap. I'll just be honest, they are very cheap. And the other is that they exist everywhere. So if we're trying to build a model that other health systems can replicate, if you're using undergrads as the substrate, you can put this almost anywhere. So what I've been trying to do for the past couple of years, and I think now have proven beyond any reasonable doubt, is that yes, you can get undergraduates to do this. You can teach them this process. You can have them use the tools we built to find treatments for patients. So these undergrads end up as the router between the scientific literature, the scientists, the physicians, the patients, and then the AI tools that we and others have built for finding treatments. They do it all. And we actually have gone so far as to build even our own EHR. And that allows us to very rapidly review all the cases that we have under consideration at any one point in time. I've also built up a wonderful faculty and staff at the Institute for doing this as well. So here's a few of the folks we have right here, so that we can run more or less any kind of experiment that a patient might need run on their behalf. And what I'll do now is just give you some examples of the kinds of things we have done with this research consultation service, so you can see what precision medicine looks like in practice on a day-to-day basis. So one of those undergrads was dealing with a patient. They had a mutation in a gene called TMLHE. She figured out this is a loss of function in this gene, so that there was only one of the two alleles, or one of the two genes was still functional for this particular patient. And this led to deficiencies in carnitine biosynthesis. So this patient was short on carnitine at this point. And it seemed logical to recommend supplemental carnitine as a therapy for the patient. So this undergrad wrote this into a research report, sent it to a physician. The physician believed her. Because she said, look, here's the logic, here's why you should do this, here's why this is safe, go ahead and try it. And they did. And so this is a patient that's suffering from lots of seizures, who now suffers from almost none by aggressively supplementing with carnitine. And then Jillian had a patient, they had a mutation in RhoBTB2. She inferred it was a gain of function in this case. And that's because the mutations occurred in a domain responsible for degradation of the protein. So by breaking the ability to degrade the protein or have it tagged for degradation, you got an abundance of this protein. So she could not find a direct downregulator of RhoBTB2. But she did find another gene that regulates RhoBTB2 called E2F1, and she found a downregulator for that gene. And so that happened to be celecoxib. And that report, which she generated, has been accepted by a physician in Brazil, who's since put the patient on. And the parents are now reporting back after a couple months that, I've met patients with this disease, and they're effectively vegetative. It's just awful disease. And these patients apparently are sort of alert and looking and focusing and paying attention to things really for the first time in their lives. And so that's big. We got to do some science to see, okay, is this real? How do we measure the endpoints here? Because there's other patients out there that we know with this disease, and we'd like to capture what's happening with them based on the experience in Brazil. And then Lindsay was working on a patient with a mutation in MAPK8IP3. She reasoned it was a partial loss of function in this gene. And when she looked it up in miniKanren, she saw right away that vitamin A, at subtoxic quantities, will actually potently upregulate this target. So you go from half the amount of the gene to closer to normal expression in the gene. The patients with this disease, they don't walk, they can't even stand. And there's not many of them, but of the ones we know of, none of them could walk or stand. But after about 6 months on very high dose vitamin A, and don't try this at home because vitamin A can actually be very dangerous if you're not careful. This is why if you eat polar bear liver, it will kill you because it has too much vitamin A. This patient actually started standing, not just standing, but also walking. So the mom started sending us these videos saying, look, my kid is actually starting to not just stand, but take steps. And so he's been taking ever more steps since, and she now feels a moral obligation to report this finding to the patients and the patient community. She's like, look, this is clearly working for my kid. I have to tell the other parents what's going on. So we've worked out a strategy for how she can responsibly disclose this to other patients and how we can help them find the right dose of vitamin A. And then I think it'll be help to just show you one example of a non-genetic disease, or there's probably a genetic component here, but we didn't find a gene for this. And that's intractable cyclic vomiting, which sometimes people just get, and it's just awful. In this case, it was a 19-year-old woman. She had multiple episodes of vomiting every single day. So she's 5'4", but she weighed 78 pounds. By the time her parents reached out to us. And they were just desperate. They said, we have tried everything to try to help her. We don't know what to do at this point. But she's permanently hospitalized. No one knows what to do. But we said, you can't have tried everything. Everything is a big thing. Because there's 30 million papers out there. 64,000 of them are on nausea. 4,000 are on treating nausea. And 374 of them, there's 374 distinct treatment modalities for nausea reported somewhere in the literature. So we said, look, you haven't tried 374 things. So we guarantee you there's stuff left to try. So we just ranked it. We did a histogram saying, okay, what is the most popular treatment for nausea? And then all the way down to stuff that has only one paper in support of it. So at the top is Zofran. It's like the most popular anti-nausea drug there is. But if you keep going down, you eventually find the region in this curve where she'd never tried it. And then we could take that region and we could rank it by safety and say, what's the safest thing in this unknown known region where it's published, but people don't just know about it because it hasn't been widely cited. And in that region, we found all the way out at 3 citations, nasally inhaled isopropyl alcohol. So rubbing alcohol on a cotton pad, and you sniff it when you feel nauseated. So we sent that back to her team. They recommended it, and literally in the same day, she came back and said, "Oh, it worked. I just aborted an episode of nausea that would have led to vomiting, and it kept working." And so when she first checked in again, it had been 8 months without vomiting, after vomiting multiple times every day for years. She was up to 125 pounds from 78. And more than that, the reason they checked in was that she got married. She got out of the hospital, she married her high school sweetheart, and they moved to San Diego. She never would have seemed possible for her. And I mean, what blows me away is that the solution was sitting in her bathroom cabinet the entire time. It was right there. And so she's got her life back, as a consequence. And there was no genetics, no molecular biology involved. It was just ranking publications and having applied NLP to the literature. That's all we had to do. And so when people reach out with more chronic conditions, we can run these queries too. We can give you a histogram of all the standard treatments and very quickly a long tail of very weird treatments. And sometimes you get very lucky in that long tail. If you're interested in observing this process, you're certainly welcome to come down to UAB on a Monday. We hold case reviews every Monday and just review every active case to see if there's an update to process. We are moving into the UAB health system. And if you're wondering whether it's made, or miniKanren has made predictions for Bertrand, well, it absolutely has. If you run Engli1 through the system now, it will tell you that he should take broccoli. Really, specifically sulforaphane, a component within broccoli. Luckily, you can get this in condensed pill form, because otherwise it would be 60 pounds of broccoli a day. I mean, it's just obnoxious quantities of broccoli. It builds a little proof tree. It says, look, this sulforaphane may treat Engli1 deficiency because when you have Engli1 deficiency, you can't activate NRF1. NRF2 can compensate for inactive NRF1, and sulforaphane upregulates NRF2. So there's a very simple logic for why you'd want to do this in Engli1 deficiency. Yet another way is actually through static analysis. If you use, there's a really interesting, I guess this is a functional programming language if you think about it. It's like a graph rewriting language. That's what this language is. It's a graph rewriting language. So it is a functional language. It's called kappa. And you can model, enzyme reaction systems using this language. And then you can simulate the effect of removing an enzyme. You can simulate the effect of removing a gene. And we've done some of these simulations to say what happens in the absence of this gene. What goes up, what goes down, what disappears, what appears. So you can use static analysis to avoid the simulation step and just predict what's going to appear or disappear as a consequence of taking out an enzyme. And people have actually done this work too. People in our field that have worked on static analysis programs have done static analysis of biological systems encoded in kappa. So these days, if you're wondering how Bertrand is, he's actually doing pretty well. He's a happy little kid, and I can't really complain. He's got 2 younger siblings who he loves to play with. He's more or less happy all day, every day. His vision is perfect. He has no seizures anymore, down from hundreds per day. And I'm optimistic about what the future holds. I think we have a lot left to do for him in this disorder as we sort of chip away at it one mechanism at a time. In fact, even more recently, he's begun using an eye gaze communication device. I guess he's been doing this for about a year and a half now, and getting better and better with his vocabulary as we engaged him, to the point where he even asked for pet fish, specifically a whale shark. But we did the best we could. So I think, I'll do one last story. I'll tell you that we do the same thing for cancer too. So we have a whole clinical trial pipeline where we genotype tumors, we look for the mutations driving the cancer, and then we look for medications that counteract the effect of those mutations. But that's just cancer. That's boring. We've had success stories where we found out that there was this guy who had intractable prostate cancer. Turns out, genetically speaking, the tumors were ovarian cancer. It can happen. It's a weird thing, but it can happen. And so, we actually had to treat him with an ovarian cancer drug. All right, so final example. So this is back to Bertrand. So last May, Star Wars Day, I took him to the Science Center in Birmingham, took him down to see the fish, and then 5 hours later, around dinner time that night, he was almost dead. He had septic shock. And so I go, okay, that's weird. I didn't even know what septic shock meant at the time. Because again, I'm not a physician. And for us was the launch of a second diagnostic odyssey. So while he's going into the ER, I'm asking miniKanren 3 questions. I said, what causes septic shock? What are the consequences of septic shock? And what are the treatments for septic shock? And it spits back albumin as one of the things way down on the long tail for treatments for septic shock. I said, oh, well, that's important because we know from the NIH studies that Bertrand has chronically low albumin. And if you have low albumin, you're going to die with septic shock. You will die. So I told the medical team, look at this paper, give him albumin. And they did. They actually listened to me. And so he got albumin, and he survived the first night. So septic shock by itself has 50% mortality. And if you have these aggravating factors, it goes up real fast. So that began week 1 in the ICU. All the cultures were negative, but he was kind of stabilizing. And by the end of the first week, he was on high flow oxygen. You can see that right here. Into week 2, slow improvement continued, very slow improvement. He got onto very low flow oxygen, and they felt comfortable discharging him on Thursday with home support oxygen with a mild fever. But unfortunately, 24 hours later, he was back. He was in extreme pain. And when we took him to the ER, we realized a lot of other stuff was now going wrong too. He had a bone infection. He has swelling in his abdomen. He had fluid outside the lung building up so fast that it collapsed his lung. And yet his cultures, looking for the organisms causing this, were all negative. So this began this sort of like rapid medical interventionism program where he had 2 surgeries to clean out the infection in his bone. He had to get a chest tube because you have to get the fluid off the lung. And this is him shortly before he was supposed to go into surgery for this. And then unfortunately, 4 hours later when he was about to go in, he coded in the ICU and came about as close as I think I've ever seen him to dead. So when we took this photo, we actually were basically just saying goodbye. I thought this was the end for Bertrand this year, this right here that night in the ICU. But I pleaded, please operate, please let's try this. At 1 a.m. I said, well, he's stable enough now. We're going to wait for morning to do the procedure. Then by 8 a.m. he was definitely not stable. So, because his hematocrit had plunged to 19%, I said, well, now his hematocrit is so low, we can't even do surgery. So he got a blood transfusion, they put in a chest tube, they put it in the wrong place, so he coded again. So he got another chest tube, and this one actually worked. So we were able to finally get the fluid off of his lungs and somewhat stabilize him. Unfortunately, the cultures were still negative. We still had no idea what was driving this disease. And this brought us 3 weeks into Memorial Day weekend. And I'm sitting there in the hospital thinking, let's try this, just one after another. Like I'm just aggressive like that. And the resident says, well, hold on a second. This is Memorial Day weekend, and nothing happens on weekends in hospitals. And this is actually true. Nothing happens on weekends. Just don't even bother going to the hospital on the weekend because nothing happens. So I said, okay, well, if nothing's going to happen, I'm going to write some code. I got to figure out what this thing is. So I created a diagnostic extension to miniKanren. And it's pretty simple, actually. It's got this knowledge graph. It knows what diseases cause which symptoms. So why not just run it in reverse? Let's say if you have some disease that explains all 4 major symptoms, put that up at the top. And so you literally just got a ranked list of all known diseases that could cause the constellation of symptoms we saw in Bertrand. And if you look at the ones that explained everything, there weren't many. there was just a handful of diseases that explained all the symptoms that we saw inside of Bertrand. So we started aggressive testing for these. And in parallel with that, did metagenomics. So your metagenome is it's all your DNA plus all the other DNA floating around inside of you. So we all got pathogens or weird bacteria in us at all times, and some of those shed their DNA. So you can see them that way. So if you take a sample from somebody and you look inside that sample, you will see the organisms, but you'll also see their DNA. It's just there. And so you can use this as a way to find what's actually inside somebody. And I wanted to know, could we use this for Bertrand to find the bug inside him? So checked his blood and pleural fluid out of the hospital from pathology. And we drove it up to Huntsville, Alabama, where there's a sequencing facility. I relayed it at a jack-in-the-box to this guy, my number 2 at the Institute, Andy Krause. He got it to this guy, Sean Levy, who runs the sequencing lab, and he did 40x shotgun sequencing 4 times over in 3 days on these samples. So now we had enough data to figure out what this thing really was. And if you look at a distribution of all the organisms inside Bertrand, oh God, there's a lot. There's always a lot in all of us all the time, as it turns out. So this was not terribly useful in and of itself. But if you intersect it with the list that came out of the AI, there was only one thing left, and that was Pseudomonas. So you wouldn't typically expect to see Pseudomonas in healthy people, but it was plausible that it had made its way inside Bertrand. So the question was, can we prove that this is what it was? And it turns out we had enough data to do that too. Because the way we ran the experiments allowed us to localize the pathogen to his lungs. What we did is we burst all the cells in the lung sample to check to see if anything changed the distribution of its genetic content before and after this bursting process. And the logic here was that if there's not a lot of something in the sample, or not a lot of DNA in the sample, then you can't see it. But if you burst the cells, then whatever is there, you're going to see it now as it's through its genetic fingerprint. So we burst the cells, and this allowed organisms that were in the sample itself to be suddenly overrepresented. Suddenly they got more of their DNA represented in the sample by bursting it. So we did this, and one organism had a massive shift only in the lungs, and that was Pseudomonas. So Pseudomonas went from 2% of the genetic content of the sample to 13% of the genetic content of the sample. And we said, well, that's clearly what this is. The other big hint was that if you looked at the viral DNA floating around inside Bertrand, there was a lot of it for something called Pseudomonas Phage, which is a virus that eats Pseudomonas. So if there's enough, if something's eating Pseudomonas, it's got to have something to eat. And so we thought it was there. There's also evidence of non-trivial amounts of E. coli. And so the drug recommended to kill both these things simultaneously was meropenem. It's a very powerful antibiotic, sometimes called gorillacillin, because it does a lot of stuff. And after 4 weeks in the ICU, he was out within 48 hours. And onto a regular pulmonary floor. And within a week, his effusions were receding, his abdomen was no longer swollen, he was back onto room air, and when we did another round of metagenomics at the very end, just looking for Pseudomonas and E. coli, both of them were gone. So there's nothing, no trace of these organisms in his system at that point. So this is him right before he went home, and this was him reunited with his fish. This is all back in May. This is June by now, actually. So he was very happy to go home and see his fish. And then 1 week later, I kept a promise to him that I'd made while he was in the ICU. This is Father's Day. We drove him down to the beach in the panhandle of Florida and took him swimming with dolphins. So this is a week after he got out of the ICU, or a week out of the hospital, he was literally swimming with dolphins. During that time, I got an NIH grant funded for a clinical trial for NGLY1 deficiency. And that trial now starts in 2 weeks. So thankfully he survived. Thankfully he'll get to try this new experimental treatment at the Mayo Clinic starting in 2 weeks. So I don't want to poach anybody. But if you've been inspired and you're interested in thinking about spending some spare time on biomedical endeavors, I do recommend this one book. This is my one recommendation. This book, you can read it in an afternoon. It tells you pretty much everything you need to know as a computer scientist to kind of understand at least the vocabulary of the field. And we do have clear immediate needs for NLP. So I want to reprocess the full text of everything with better, more modern NLP to build better knowledge graphs, really ultimately in the service of building proofs for patients as we make recommendations for them. So the take-home message here is that I really don't believe there is anything that is not actionable, whether it's in medicine or otherwise, because I think we can. It's certainly always been the case, no matter what anybody told you, that in the event of the non-actionable, in the event that there's nothing you can do, you can always do science. So thank you, and if you have any questions, I am more than happy to take them.